My name is Henry Childs. I'm a third-year PhD candidate in the Donald Lab, and I'm really excited to talk to you all today about computational protein modeling. There's going to be a number of things I'm going to cover in this talk. The first is going to be I'm going to give a brief overview of computational protein design, and this will be just a generalized definition of the workflow, common objective, and we'll be talking a lot about binding today specifically. I'll also be talking about predicting binding affinity in our in-house software suite, Osprey. It stands for Open Source Protein Redesign for You. It's a free and open source protein redesign software. And I'm going to be talking about a specific algorithm in that software suite called K-star, which is for approximating binding affinity. I'm going to specifically, once I introduce that algorithm, talk about certain assumptions we're making and simplifications when we're using this type of algorithm. And then I'm going to close talking about why provable properties are valuable in computational protein design. There's also a number of things I'm not going to cover in this talk for the sake of brevity. Hey, good morning. First I'm going to not cover is K-star proofs for energy bounds on conformations. This is a really valuable and interesting topic, but this is a talk that I've and other people in the lab have given before. We have video lectures online and lots of publications about this. I'll also not be talking about accelerated binding prediction algorithms. So these are things like branch and bound K-star. These are accelerations to the K-star algorithm. I'm also not going to be talking about diverse design objectives such as positive and negative design. This would be things like predicting off-target effects, dipeptide design, resistance mutation. I've also nixed solvent modeling for the purpose of this presentation for simplicity. So generally the idea is that for this presentation, we're going to be considering a specialized, simplified model for a special case, but know that we can investigate arbitrary design objectives given these fundamental principles. And Osprey has been used in a number of applications. It's currently produced designs that are in 32 clinical trials, and there's a number of publications I recommend reviewing that deal with all different kinds of systems. So let's jump into it. So what is computational protein design? Generally, it's the use of in silico methods to design proteins with novel functions or properties. One of the earliest and my personal favorite examples of this is from 1997, where it was a completely rigid model de novo design protein was developed to target a zinc finger domain. So everybody has their favorite way to do computational protein design. It wouldn't be possible to talk about all of them in a quick 30 to 40 minute lecture. So I'm going to generalize this into a three-step workflow that'll kind of persist throughout this talk. The first is we have our biophysical model. And this definition I've left intentionally very vague. This could be a atom-atom contact representation matrix. It could be a pairwise energy matrix. It could be any sort of representation of your chemical system. Then there's the search. This is what I would call the algorithm. This is when you're actually taking that input of your biophysical model and doing some sort of computation on it. And then from there, typically you want to have some sort of experimental validation of your designs. Also, one more note about that middle section, step 2, that algorithm. A lot of you all might be familiar with this in a sort of two-step process. A lot of times you'll have some sort of desired target, you'll develop a backbone that's amenable to that target, and then you select sidechains from that. So that's also compatible with this sort of generalized three-step workflow for computational protein design. So to put this a little bit more formally, let's consider that we have a design A and a biological target B. A very common objective, and the one I'll talk about today, is determining binding of two proteins in silico. So given in silico design A and biological target B, find both sequences and conformations for A and conformations for B such that A binds B. Note that we're looking over shape for both, but specifically identity for A, obviously because we wouldn't be doing mutations to a biological target. So this is a really valuable question to have an answer to. If we have a known disease pathway that we can inhibit, it's really valuable to be able to design a very potent inhibitor, for example. But computationally, it's very, very expensive. So our approach in the Donald Lab is to do something called the K-star algorithm. I'm going to start this section with a claim. And that claim is if we have exact partition functions, we can compute the equilibrium binding constant, that's Ka, for our system. So let's say we have protein A and protein B binding to form the complex AB. That Ka is going to be the concentration of our complex times the standard concentration, typically 1 molar, divided by the concentration of both the reactants. So some of you all might be thrown off a little bit by this definition, by that C in there. I won't perpetuate this throughout the talk. I've just put this in there as a reminder that all of these equilibrium constants are dimensionless, so there's no units on these. So later on in the talk, if I am showing a ratio and I say something like this is dimensionless, generally because there's some sort of implicit standard concentration there.
Can I ask a quick question?
I'm going to run through the presentation,
yes.
and I'm happy to answer as many questions as we have at the end. The second is our equilibrium condition. We know that if we have the chemical potential of A and the chemical potential of B, this will equal the chemical potential of our complex. And then last but not least, we can define the chemical potential as the negative Boltzmann constant temperature, natural log of our partition function divided by the number of particles in our system. This is the standard definition of chemical potential for a system of non or weakly interacting indistinguishable particles. So this is all pretty fundamental chemistry. What happens is quite interesting. If we plug equation 3 into equation 2, we get this kind of confusing looking thing, but if we pull out all of the constants, get rid of the natural logs, we see that the ratio of partition functions is equal to the ratio of the number of particles. Keep in mind though, we're assuming constant temperature and volume, so we can very trivially rewrite this as the ratio of the concentrations, which is simply just Ka. So this is the theoretical, fundamental theory behind a theoretical approximation to Ka. So this looks pretty straightforward, but the problem is computing these exact partition functions isn't really feasible in reality. The system, an ensemble could have an infinite number of states. So a lot of times what we want to do is we want to systematically sample this type of ensemble. And I'll talk about how we do that a little bit later. Know that a lot of times though we'll approximate these partition functions. I'll call Q the approximation to Z. And that is simply just a sum of the Boltzmann factors for all the states in the system where Q is approximating Z. So from equations 5 and 6, the K star score is the ratio of our approximation of the partition functions, which approximates Ka. Also note another point of common confusion. K star can mean two things. It can mean K star score, which is this ratio here. It can also mean the search. I'll try my best to say K star score and K star search to differentiate. So to make these approximations, we definitely need to simplify our biophysical model. So we have our reality. This is what would happen if we actually went in a lab, ran something like BLI and looked at binding affinity. But we want to approximate that through some sort of in silico method. So we build an ensemble, which is some sort of physics description of this reality. And there's a number of things we can do with this. I'm going to propose two. The first is we could exhaustively enumerate this entire ensemble, which would be very computationally intractable. In fact, computation of exact partition functions is still an open problem. So instead of doing this, I propose that we actually have some sort of scheme to systematically sample a sub-ensemble from this ensemble that we built, which would be some sort of approximation to Ka. Note that when I say ensemble, I'm not talking about something like some sort of stochastic sampling. We actually have a way to do this, which I'll talk about in depth later. So we actually have some sort of guarantees about our search. So one thing I want to note specifically is actually state the philosophy for a lot of our designs, because it can sometimes throw people off. The claim is not that we are building a model that has the best physics. The claim is that we are building a model that is physics tractable for a large complex. And then we can add more details as algorithms improve. So this review study from 2016 really drives this point home. Let's look at the y-axis first. On the y-axis, we're looking at the quality of the biophysical model. So at the very bottom here, this SC, this would just be some sort of rigid model. So no flexibility, no mutations. This is quite tractable. If we look at the top of the y-axis though, this is a very high quality biophysical model. This would be something with explicit quantum mechanical terms, mutations, continuous backbone flexibility, continuous sidechain flexibility, really good binding affinity approximation, multi-sequence design, everything we like. But we want to contrast that with the x-axis, which is actually the design size and what that is tractable on. So what we're seeing here on this red line, this is the computational power pre-2016. The blue is that computational power in 2016, and those numbers are references to specific algorithms that this figure was pulled from. Note though that when we're talking about binding, we're really talking about this area over here, which is a protein complex. So simply doing quantum mechanics on a large protein-protein complex generally isn't very feasible, and that's what this figure is really showing. So generally, if we're going to calculate an approximation or a partition function, there's really two components, and that's what we're talking about for the rest of this talk. The first is the set of conformations in that ensemble, and the second is the method used to score that ensemble. This is not just the energy function, as it's typically called, but also what we're doing with those energies. There's also four main takeaways I want you to have from both of these simplifications we're going to be talking about today. The first is what we're assuming in our tractable model, what we stand to benefit for our assumptions, why we're doing it in the first place. Reasoning behind our assumptions, why we think we're not just arbitrarily making decisions and pulling quality of our model, how we're doing that systematically, and then how specific algorithms can help improve our model. Go further at that y-axis on that figure. So the first implication is that residue conformations can be represented as minimized, empirically based rotamers. This is typically called a rotamer library. So the general idea behind a rotamer library is mine the PDB, so mine known crystal structures, high quality crystal structures, and look at the dihedral angles for each side chain and graph them. So what we're looking at here is just an example of isoleucine, and this is taken from a number of high quality structures in the PDB. If we actually graph their dihedral angles, we'll see that they actually have a grouping. If we were to assign, through a systematic statistical method, groupings for each one of these, we could then take the mode, and that would be our rotamer. So rotamers are empirically based, low energy conformations of side chains. We then repeat this process for the other 19 amino acids. We can actually build up a library, and that's how we're significantly cutting down our conformation space. We then minimize during our search, and we can represent rotamers with something like this. So let's talk about the assumptions we're making when we're doing something like this. The first is that residues only occupy dihedral angle modes. But the benefit of this is that our search space is significantly more tractable, and we can design on those large systems. So keep in mind that graph that we were looking at earlier. Third, it's also worth noting that generally rotamers can lead to better designs because there's actually an empirical basis for a lot of our side chains. If we just simulate something in silico, something that's very constrained for a long time, it may or may not have any sort of empirical basis for actually adopting that conformation. Some of the reasoning why I think this is a pretty reasonable approximation, the specific rotamer library that we use, it's based on 240 high-resolution crystal structures. It does have 153 rotamers in it, so it's not like we're using one rotamer for each amino acid. We're still sampling quite a large conformation space. Also, we expect, based on the statistical distribution we see when building these rotamer libraries, we expect some of those non-sampled residues to occur more rarely. And as usual, algorithms can help with this. So the first is what I would typically call rotamer +1 or wild-type flexibility. If we have a desired target, we have algorithms that can read in that structure, and if it has rotamers that aren't present in our library, we can dynamically read those in, and we can search over any sort of unknown or unaccounted for rotamers. The second is a much bigger algorithm. There's a number of publications on these. I think they're really, really interesting. This is called minimization-aware rotamers. So everybody does minimization. You get the structure, you minimize it, you do a search, you minimize it again. The key here is that we're actually minimizing while we're doing our search. And let me visualize exactly what I'm talking about here. So what we're looking at in this top figure is 3 interacting residues. And you can see we have this overlap here representing some sort of unfavorable energy, and depending on our parameters, it could be a steric clash, so ostensibly infinite energy. If we only do a rigid rotamer search, how do we know that if we minimize this, it wouldn't potentially be one of the lowest energy structures? So the key here is that we're actually doing this minimization during the search. And generally, this almost always leads to lower energies. We're looking at here on the y-axis is energies going down. This x-axis is a number of PDB entries. The yellow is rigid, and the blue is minimized rotamers. Jumping over to the second assumption. The second assumption is that the protein backbone is rigid for a lot of these very simple models that we're doing in Osprey. So one of the benefits is this significantly reduces the complexity of our search. Something to note is I've just talked about how complicated it is to do a sidechain search. Keep in mind the sidechain is attached to the backbone. So if we do a rotamer search, and we move the backbone even just a degree or a couple degrees, we then need to re-perform that backbone search. So backbone flexibility is really, really expensive for these types of approaches. What point of reasoning, our backbones are empirically based. We're often very common sampling from the PDB. And especially when we're doing really, really, really large designs, we expect that doing just a few mutations will hopefully not induce large backbone movement perturbations. As always, algorithms help. One of my favorite I use all the time is translation rotation. So the backbone remains rigid, but if it's in an active site, we can translate and rotate it to look at different conformations of that design. There's also algorithms such as deeper and cats. So keep in mind I'm saying we're looking at a very simple model, but we do have models that can do continuous flexibility. For example, if you were designing in a region that had a flexible loop, I would definitely recommend doing some backbone flexibility. The key here is that oftentimes there's sometimes trade-off between sidechain flexibility and backbone flexibility. So those really kind of go by a case-by-case basis. Lastly, some of my own work with Nathan Guerin and Pei Zhao. was working on parallelizing a search across multiple different backbones by sampling from the PDB, and generally this increases our sequence recovery and improves our predicted binding fidelity. Jumping over to component 2, this is our method of scoring for our approximated partition function. So the simplification is that our conformations can be sufficiently scored and ranked using a pairwise interatomic potential. Generally what this means is we're using molecular mechanics. What we're looking at here is just a visualization of the Ambertool's energy function. One of the assumptions we're making when we're constructing and doing a method of scoring like this is that we can accurately approximate the partition function by sampling from the conformation space systematically. So one of the benefits, as we talked about in one of the earlier slides, is we can just sample a subset. Generally, there's sort of a thermodynamic basis for this, is that in Wilson weighting, these high energy states generally contribute negligibly to the partition function. So let's look at that visually, because that can be hard to kind of visualize. So if we're doing a search and we have all of our minimization aware rotamers, the general idea here is that if we have something that is at a very high energy, we can spend very little time refining the bounds of the energy because they contribute so negligibly to the running sum, which is the partition function. So we might spend a lot of time in this region once we have good bounds, refining those bounds, but for structures and conformations, at a higher energy, we don't have to spend as much time on those because, generally they won't contribute much to the partition function. Won't talk about pruning today, but that's the general idea. And as usual, algorithms help. One of my personal favorites is BB-K-star. This is branch and bound K-star. Sort of extends some of the philosophy about searching over conformations to sequences. It's one of the few optimization algorithms I'm aware of that could be potentially sublinear in the number of sequences, which is pretty exciting. Also, we have a number of K-star algorithms for specific design objectives. So if you're doing just one or two mutations, energy window approximation of K-star is good. That's EWOC. And then we also have minimization aware K-star. That's also a great algorithm to look into. Let's go into our last assumption. The last assumption for this component is that the protein-protein complex can be represented as a sum of pairwise energies. So the main benefit is that we can really, really quickly compute, evaluate, and prune conformations. So generally, we can start with our rotamer library. We can very quickly construct a pairwise energy matrix and then search over that. Also, we can vary our degrees of freedom. This is really something we don't want to underestimate. There's oftentimes a trade-off between how many bodies we have in our energy function and the amount of flexibility that we can look over. So oftentimes there's sort of a balancing act there. Some of the reasoning behind this is, as we talked about earlier, we simply can't design with a higher quality biophysical model for some of these search spaces that we want to look over. And that the specific Ambertool's force field that we look is parameterized specifically for proteins. Per usual, algorithms can help. It's worth noting that the K-star search is agnostic to energy function, which is pretty cool. So we've written this to be very modular. So if you have a very small design space with just a handful of residues, you could plug in a much more complicated many-body energy function and have some trade-offs there between the amount of flexibility you're looking at and the amount of interactions in your design. But there's, of course, a trade-off there with flexibility. So those are, again, sort of on a case-by-case basis. Lastly, some other exciting things here. We can do something like a hybrid optimization machine learning search. Some of my favorites are by Mark Allen. Both Epic and Lute are really exciting algorithms for sort of learning and doing fittings of energy minima, and those can help accelerate our search. So we've just talked a lot about the components of our assumptions and what assumptions we're making. The question is, why do we want to simplify in the first place? The answer that I would propose is that provable properties are really desirable. The first advantage of a model that has a search with provable properties is that K-star can be shown to approximate the model's true partition function. Think about if we weren't searching systematically and having a scheme to actually enumerate a subensemble. If we just randomly sample from a very large conformation space, there's a high chance that we're going to get lots and lots of high energy structures. We don't really have any guarantees that we're approximating the partition function, which is very valuable. The second is that we can actually isolate our issues to our biophysical model and not our search. So what that means exactly is that we can actually feed back information from our experiments into our biophysical model. One of my favorite examples of this was just recently, a non-canonical lysine rotamer. I won't read that out, but a non-canonical lysine rotamer. They were doing designs for non-canonical binding. They had an in silico method to sample rotamers. When they solved the crystal structure, they saw that the side chain conformation in the experiment was different from what Osprey predicted. It then inserted that rotamer back into the biophysical model, reperformed the search, and Osprey selected that same rotamer that was present in the crystal structure. So this is one of the many, many ways we can feedback experiment into our model. And then last but not least, it can be very valuable because oftentimes we talk about making these algorithms faster, but the bottleneck can sometimes be experimentation. And if we want to test a number of peptides, it could cost $60,000 in 3 to 6 months of manpower to actually do those. And so oftentimes, a lot of times in the lab, we'll do some sort of in silico validation. We can actually tune our biophysical model before we experimentally test everything. An example of this I really like is from 2012. There was a design for a Cal-P PDZ domain. There was a number of known binders, and they took those 12 known binders, tuned the parameters of the energy function, which were part of the biophysical model, performed a search, and had a number of successful binders. So that is a good way to do in silico validation. We can also do things like sequence recovery before we test anything in the lab, and also we can look at alignment RMSD to known structures. I'll close by saying if any of this sounds exciting to you, Osprey again is free and open source. It's available on our GitHub. We also just recently published 2 different protocols. These will actually walk you through step by step. Here's how you download the software. Here's how you download Java. Here's how you run our searches. This protocol here, I've recorded over an hour of videos. There's a lot of tutorials here if you're interested. And then this is another quick tutorial. Previous Donald Lab member Nathan Guerin about predicting resistance mutations using Osprey. Also, feel free to reach out. My email is henry.charles@duke.edu. And I'll just close by saying, here's some citations, references to those aforementioned K-star algorithms. I think these are all really exciting. I recommend you look into them. It's been great talking with you all, and I'm going to open up the floor to questions.